is a friendly collaboration between two Sri Lankan communities from the two countries separated by the longest but the friendliest border in the world.
With every challenge comes an opportunity. While COVID-19 brought life as we know it to a halt, it also opened up many opportunities and gave everyone the gift of time. For those who long to spend time with their families, they got all the time in the world. Those who disliked long commutes to work got the opportunity to work from their homes.
Here we have an academic situation, everybody learning something. So I wish we have more seminars and webinars like this.
First, I want to thank the Alumni Association of the University of PERAHERA, Ottawa chapter in Canada for organizing this webinar.
Taken care of hundreds of thousands of patients in this country as well and world over. So hats off and salute to all the healthcare workers.
As Shirani mentioned, that I have done research on HIV for a long time, and I had to be careful when I say I come from the HIV world. Last time I said that, and somebody texted me and saying, "Hey, I didn't know that you had HIV." So I said, "No, I do HIV research."
I'd like to extend my sincere gratitude to our heroes, the doctors who put their lives at risk to save others and their families at risk as well.
A warm welcome to everybody and greeting from Ottawa, Canada. I think we are having participants from various countries in various different time zones. So good morning and good afternoon and also good evening to all. And I am PERAHERA, Secretary of the Alumni Association of University of PERAHERA, the Ottawa chapter. And this is the second webinar that we have organized. And the topic this time is the new COVID therapeutics and the very, very important topic and timely topic. The therapeutics and vaccine. And I don't think that topic needs any introduction or any clarification. That has its own credentials these days. And the whole world is looking for the therapeutics and vaccines. So I'm sure this topic is going to be in the limelight for next couple of months, if not for next couple of years. So this time, I must mention that we are very happy that we organized this webinar in collaboration with the Sri Lanka Foundation in California, in the States. And they were very, very helpful to organize this and also introduce two doctors to make their speeches. So let me recognize a few personalities for our webinar today. First, I have the honor to welcome the Sri Lankan High Commissioner, Her Excellency, Madam Sitara Khan. And thank you, Madam, for joining us. It's a great strength for us, your presence. And also our two doctors, they are veterans in the field of therapeutics and vaccines, COVID therapeutics and vaccines. So Dr. Deepthi Jayasekara and Dr. Dushyantha Jayaweera. And thank you very much for joining us. And it's before start the presentations, and I think I should read a few housekeeping rules and also the disclaimer. I think I will read them. The first one is the organizers are recording this webinar. So therefore, we request attendees not to record any conversation. And later on, the recording will be posted over our website. So you will have the access to listen to it again. And also be respectful, keep your comments helpful, and also considerate of the host and the other participants. And during the questions and answer session, you may ask the panel of doctors any questions you may have through the chat line seen at the bottom of your screen. The moderator will direct the questions to the participate speakers. Thank you for your participation. And let me also read the disclaimer. I think this is important for a webinar of this nature, because there are technical contents that will be presented. So this webinar is designed to provide helpful advice and information on the subject of the novel coronavirus called COVID-19. Individual diagnosis and concerns should be directed to each participant's personal physician. The authors and hosting company are not responsible for any specific conditions that may require medical treatment and supervision, and are not liable for any damage or neglect consequences arising from this webinar. Please be mindful of this disclaimer. So now I have the pleasure of inviting the president of the Alumni Association of University of Peradeniya, Dr. Nimal De Silva, to give us an introductory speech. Please, Nimal.
Thank you, Dr. Anugraherath, our secretary and the master of ceremony today. Good morning, Her Excellency, our acting Sri Lankan High Commissioner to Canada, Madam Sitara Khan, and good morning to Dr. Walter Jayasinghe and two doctors, our experts, Dr. Deepthi, Jayasekara, and Dushyantha, Jayaweera. Good morning, good afternoon, and good evening, everyone elsewhere in the globe. On behalf of the Alumni Association of University of Peradeniya, Ottawa chapter, it is with great pleasure I cordially welcome you all for the webinar today on a very timely topic in the middle of our great optimism about battling the pandemic. As the president, I like to take a minute to tell you a few words about who we are and what we do. So we have these are executive committee with the representation from 9 faculties. And our association was founded in 2011, less than almost 10 years ago. And membership has grown to nearly 70. So with thanks to all our dedicated members and executive committees in the past, under our past presidents, Dr. Nimal Ratnayaka, Dr. Sange De Silva, Dr. Dhammika Herath, we have come a long way in terms of reaching our goals, objectives, mainly foster friendship among alumni and Sri Lankan community in Ottawa. That was our original objective. However, it's a great pleasure that today we have been able to extend that friendship to other Sri Lankans as well as other non-Sri Lankan friends throughout the globe with this seminar. So also our second objective is actually equally important, help University of Peradeniya, and not only the University of Peradeniya, all the other universities whenever we can in Sri Lanka, and other educational programs for the benefit of Sri Lanka. So these are a couple of ongoing and past activities that we have been doing, offering scholarships, special financial supports for needy students, and helping the newcomers when they come here, then donate research equipment, books, computers. Then our members conduct short courses, seminars at University of Peradeniya and other universities. So to do this, our funds come from a generous donation by our members and friends in the Sri Lankan community in general. So, and also from sponsorships, funds raising through social and cultural activities open to the community. What are our fundraising and social activities? We have about 5 social activities. first thing, in the coincidence with our single Tamil New Year, we have our Spring Festival participate. These things are participated by nearly 100 to 200 community friends. Then we have, in spring, we have a Paduru party, PERAHERA G Padura, dedicated to legendary singers in Sri Lanka. Then we have a summer picnic, the health walk, and also our premier event, Hanta Night, with rich cultural show. So unfortunately, this year, all our activities had to be blacked out. So we could not do any fundraising activities or anything like that. So, but we have our determination. The life finds a way, and there's always a way. So how do we approach that? We started thinking about organizing webinars to help our members as well as participants from anywhere in the world. So as our first attempt, in October 18th, we had our first interactive webinar on why we need to control stress levels and ways to do it with Dr. Lal Fernando, another PERAHERA alumnus. So if you like to listen to that webinar, you can always go to our website, and the link is there. Then today, we are very fortunate to be able to offer our second webinar. This is very, very special location that we all should be very proud of, because it is a friendly collaboration between two Sri Lankan communities from the two countries separated by the longest but the friendliest border in the world. So I like to thank Sri Lanka Foundation for helping us with this and agreeing to collaborate with this particular project. I particularly like to thank Dr. Walter Jayasinghe, who is the founder of this foundation. And it is a great honor honor to us that he's here personally to speak few words for us. And also I like to thank our speakers, the experts in the area, Dr. Deepthi, Jayasekara, Dr. Dushyantha, Jayaweera. As you know, Dr. Deepthi Jayasekara is from California and Dr. Dushyantha Jayaweera from Florida. They both are hotspot in the US. So you can imagine as frontline doctors how busy they are. Yet when we approached Dr. Deepthi Jayasekara, within less than 24 hours, he very, very enthusiastically responded to us, even with the title right away, that he is willing to offer this with his colleague. We very much appreciate that effort. Then I also like to thank our very, very friendly cooperative coordinators from Sri Lanka Foundation, Program Director Keshani Vijay Gunaratna, and IT Specialist Chamod Amarasinghe. From our side, I like to thank the webinar number 2 committee, Coordinator Dhammika Herath, and Master of Ceremony, our Secretary Anura Herath, and with the help with web graphics, Deepani Vaidyaratna, and Zoom tech support, Sampath Nayak and Manoj De Silva. As we always say, last but not the least, we should appreciate our generous support from our sponsor today, Erika Ushlianage from Mortgage Alliance. And if any one of you willing to donate to our scholarship fund, please, you may contact aaupoc@yahoo.ca by email or send an e-transfer. We very much appreciate that. And thank you all for your participation and interest. And hopefully we see the end of the tunnel with the battle of COVID-19, as you will hear from our expert today. There is always a way. Hope you all will find this webinar useful and valuable. And stay tuned for our future webinars. We will let you know. Then stay safe, healthy, and happy, particularly be optimistic. Thank you very much. With that, I invite our Master of Ceremony to proceed with the rest of the program. Thank you.
Thank you, Nimal, for that brief but very lively introduction. And Her Excellency, Madam Khan, I have the honor of inviting you now to deliver your greeting message.
Thank you. Dr. Nimaldi Silva, Dr. Jayasekara, Dr. Jayaweera, our host, Mr. Hera. Greetings to everyone. It is an immense pleasure to address the webinar hosted by the Alumni Association of the University of PERAHERA, Ottawa Chapter, Canada, in collaboration with Sri Lanka Foundation, California, on an important topic titled New COVID Therapeutics and Vaccines. At a time when the entire world is grappling with the impact of the COVID-19 pandemic, the discovery and the introduction of a vaccine has brought immense relief to everyone across the globe. The COVID-19 pandemic has wreaked havoc on the health and well-being of people, impacted economies, and brought about untold changes across the world. Unlike natural disasters or diseases that affect certain countries or regions, COVID-19 took over the entire world almost at the same time. From the changes to the school system, to workplace environment, to shopping, to social interactions, everything has undergone a sea of change, leading to a new normal way of life. However, with every challenge comes an opportunity. While COVID-19 brought life as we know it to a halt, it also opened up many opportunities and gave everyone the gift of time. For those who longed to spend time with their families, they got all the time in the world. Those who disliked long commutes to work got the opportunity to work from their homes. In short, it paved the way for creative and ingenious ways of how we managed and lived our lives. But like many things in life, the toll of the virus has taken on people, leading to a massive human cost, both in terms of infections and loss of precious lives, posing a huge challenge to frontline workers, hospitals, and the need for medical equipment, or have all added to this challenge. The world has gone tired with the restrictions and the unprecedented challenges the pandemic has posed. The approval granted to the COVID-19 vaccine has brought about much needed hope and relief to every single person on this planet. I would like to take this opportunity to thank the PERAHERA Ottawa chapter for the immense amount of work they have done for the welfare of the University of PERAHERA. As an alumni of University of PERAHERA myself, I am heartened to see the immense contribution of all the members and the supporters and sponsors for the worthy causes that the chapter has been pioneering and has been assisting. So hats off to the team, and I hope your duties and your contribution will continue, while making way for the eminent medical professionals who are due to address the webinar today to share details and insights into this important discovery and the implications of the vaccine. I would like to conclude by thanking Dr. Nimaldi Silva, President of the Alumni Association of the University of PERAHERA Ottawa Chapter, and his team for this timely and needful webinar. I wish the organizers of this webinar the very best and good health to every participant. I thank you.
Thank you, Madam. And your greeting message. And please ensure that your presence is a good strength for us. And this kind of activities are strengthened and enlightened, and also given the due recognition by having the government representative here with us. Thank you so much. And also, while thanking again the Sri Lanka Foundation, and let me now invite Dr. Walter Jayasinghe, who is the founder of the Sri Lanka Foundation, to tell us about the Sri Lanka Foundation. Please, Dr. Walter Jayasinghe.
Well, first and foremost, I thank every one of you for participating and also thank the greatness of the organizers to think of this at this time, because medical area is an area that has not been discussed very much in our associations globally. It's always been social, entertainment and picnics and that kind of thing. But here we have an academic situation, everybody learning something. So I wish we have more seminars and webinars like this. Talking about the Sri Lanka Foundation, it was founded in 2003. We have been holding mainly social functions, and particularly important function has been the awards. I'd like all of you to know that anybody in your community who deserves an award for academic or commercial success should be nominated for awards, because Sri Lanka Foundation has been awarding over the last 18 years awards to prominent Sri Lankans outside of Sri Lanka who should be recognized as contributing towards their own profession, as well as giving pride to the Sri Lanka as a country. Please go to our website and read about our awards. Secondly, we have Sri Lanka Day once a year, which brings in thousands of Sri Lankans to a city called Pasadena. And we have a parade representing almost all aspects of Sri Lankan culture. And it is nice for you to go to YouTube and watch Sri Lanka Day presentations, as well as other presentations that Sri Lanka Foundation has. That is a short history of the foundation. I wish you all the best, and that's it for now. Thank you.
Thank you very much, Dr. Walter Jayasinghe. I think you have a great institution, and we are very happy to learn about that institution. I'm sure we will get into your website and learn further. So now I think we have come to the point where all of us were waiting. So let me call upon the Chief Operating Officer of the Sri Lanka Foundation, Shirani Stanislas, to introduce our great two speakers, two doctors. Thank you. Please, Shirani.
Good morning, good afternoon, and good evening. My name is Shirani Stanislas, the Chief Operating Officer of Sri Lanka Foundation. First, I want to thank the Alumni Association of the University of PERAHERA, Ottawa chapter in Canada for organizing this webinar. Today, I have the honor of introducing two eminent Sri Lankan physicians who will be addressing you on new COVID therapeutics and vaccines. First, Dr. Deepthi Jayasekara, who is the Medical Director of Sri Lanka Foundation. Dr. Jayasekara is a board-certified infectious disease specialist and clinical professor at Western University, Chief of Infectious Disease at Eminent Health and San Dimas Hospitals in California, dealing with a broad array of infectious diseases. Dr. Jayasekara graduated with honors from Colombo Medical School in 1992. He completed his residency at Colombo University Hospital, Sri Lanka. Having more than 28 years of diverse experiences, especially in infectious disease, Dr. Jayasekara cooperates with many other doctors and specialists in medical groups. He is also involved in many charitable projects in Sri Lanka, in collaboration with Sri Lanka Medical Association of North America. Our next speaker is Professor Dushyantha Jayaweera. He is a professor in clinical medicine at the University of Miami. Currently, he oversees the infectious disease clinics at University of Miami, Miller School of Medicine, and Jackson Health System, where over 3,200 HIV-infected patients are managed. He has been a PI and co-PI, I believe it stands for Principal Investigator, in many NIH-sponsored studies in infectious diseases, drug abuse, and site PI of many industry-sponsored infectious disease studies. He is also a member of the Center for AIDS Research, as well as a senior investigator at the Clinical and Translational Science Award, both NIH funded. He has also been a site PI in over 80 clinical trials and published extensively on HIV and hepatitis C. He was the Executive Dean for Research and Senior Associate Dean for Research at the University of Miami Miller School of Medicine. He's one of the lead investigators in the Miami Clinical and Translational Science Institute. Furthermore, he's a member of the Executive and Steering Committee of the One Florida Research Consortium at the University of Florida. He's also a member of the Center for AIDS Research at the MSOM and the site PI for NIH-sponsored IMPAACT, which stands for Improved Physical Activity After Cancer Treatment, grant for HIV. He's the medical director of Phase 1 Clinical Research Unit at University of Miami Towers, where most of the stem cell studies are conducted. Thank you all, and a special thanks to our doctors. Now I hand the webinar platform to our moderator, Dr. Jayasekara.
Thank you once again for joining us. Good morning, good afternoon, good evening, everybody joining us from the US, Canada, and rest of the world. This is a very important and a pivotal time for all of us, because now at this juncture, we are talking about not just COVID-19 and the chaos, the infection and the pandemic caused in our lives, but also the excitement of a vaccine development. So we're going to dive right into that after going through some of the preliminary slides like this. As we all know, we are going completely viral with this pandemic. This is the worst and the mother of all pandemics, as you know. This kind of pandemics happen once in a century, once in a lifetime. This particular virus, it looks beautiful here, but it's a ruthless virus, as you know. But the virus presents us a spike protein right on the surface, as you can see. That is a common target for vaccines. So we're gonna go into details later on. As we all know, coronaviruses have been around for centuries now. And these are single-stranded RNA viruses, and they are found only in birds and mammals. And there are 2 alphas and 2 betas that cause human disease. But SARS, MERS, and COVID-19 are zoonotic viruses originated in animals, right? So basically, we believe that all these originated in bats. So bat-originated zoonotic viruses are very, very virulent, very, very deadly. And so far, there have been big numbers. You've read the news all along. We've had about 300 deaths in the United States itself, and 1.6 million people, succumb to COVID-19. Unfortunately, this is rampant all over the world. So only exit strategy at this point is the vaccine. So, before going into pathogenesis, I'd like to present you with this cartoon. As you know, this is the RNA virus that has a great resemblance to or similarities to HIV virus. So HIV virus attaches itself to the CD4 cells, right? It's a T cell kind of a virus. And this is also a T cell virus. And this one attaches to the human cells with ACE2 receptors, specific receptors called ACE2. And there are some areas which are dense with ACE2 receptors, such as the lower respiratory tract and the lungs, epithelium, the alveoli. So once it attaches itself, it enters the cells and then binds or fuse with the envelope and goes through the normal process of translation and then viral assembly, viral release. So once the virus is released to the body, the virus is attacked with 2 mechanisms, all of us, right? We have T cells attacking the virus and we have B cells attacking the virus. we are mainly focusing on B cells at this point because it's the B lymphocytes that produce neutralizing antibodies. It's as simple as that. Those are mostly IgG type of neutralizing antibodies, but T cell also plays a major role in combating the virus and then remembering the virus if you get exposed to it, or remembering the vaccine if you get exposed to the virus again later on in life. So T cell through memory, T cell killer cells, and then B cells through antibodies. So yes, we get it. We are all sick and tired and fatigued with this pandemic. This pandemic is raging through the world once again in their second wave. We are all sick and tired of quarantine. We are all sick and tired of washing our hands and distancing and canceling our parties and canceling our gatherings and all kinds of things. But at the end of the day, we should ask ourselves, are we doing the right thing? How do we return to the normalcy? There's a way out for this, folks. Unfortunately, that way out comes with a lot of effort through a dedicated scientist work. And so far, within a matter of 9 months, the scientists have put together some great vaccines we're going to talk about. But vaccines will give you protection 90-95%. That's still about 5 to 10% of the people who would escape this protection. Unfortunately, the culture should continue next 6 months to 1 year. Why? Because there are some people who could potentially develop this infection even though they were vaccinated, so 5 to 10%. But other thing is, even though you're vaccinated, you can potentially develop a asymptomatic infection that we will never diagnose, because if you're asymptomatic, if you don't have symptoms, you will never get tested. So based on those two alone, I would strongly recommend to continue to wear a mask, continue to wash our hands. This is there for us to stay. Continue to social distance to the best of ability you have. And self-screening with temperatures, monitoring symptoms, those things should go on for the next 6 to 10 months. Change of culture and behavior is very, very important, especially we are experiencing an enormous surge in California right now. And we know why. We just had Thanksgiving, a very, very important Thanksgiving weekend. And there were, in spite of all the CDC warnings and recommendations, there were so many parties and stuff. So we got exposed, we know that. And our hospitals are completely inundated and we are completely swamped. I actually, for the first time in my life, I'm completely feeling helpless at this point. Hundreds of patients to be taken care of by us. We have had 5,000 cases already this year among my group mates of 6 infectious disease specialists in my group alone, but in my area alone. But think about all the other areas in this country. So we are completely upside down. So please help us by taking this vaccine. We are here to educate you. So when you talk about vaccines, generally we talk about COVID-19 vaccines at this point. But please, ladies and gentlemen, do not forget your influenza vaccine this time around, because we don't want a twin-demic. Please don't forget your pneumococcal pneumonia vaccine. Everybody over 65 should get their pneumonia vaccine, Pneumovax, and Prevnar 13. We have the system in place for that. There's a due process for all these vaccines. And then another respiratory virus will be a pertussis vaccine. That's given every 10 years. So we're going to talk about, before we talk about the vaccine, I like to present the 2 formulas that I always talk about. DREAM for preventing COVID-19. This is very well known, but I just put it together, courtesy of Sri Lankan physicians. Distance, D for distancing, R and E for respiratory etiquettes. 3, aseptic technique, meaning hand washing and hand sanitizing. 4 for masks. And I've added mRNA vaccines to that. And in order to treat, once you get the disease, once you get the infection, 80% of you all will be at home. You'll be doing fine, but you still have to take some precautions in order to protect yourself from complications and protect others from getting the disease. But about 10 to 20% will have to go into the hospital. You will have to seek medical advice. Unfortunately, at that point, we have to admit you if you are experiencing shortness of breath. So therefore, DRAPE protocol or DRAPE formula is very, very important for all the physicians to use because it's very complicated now. We have so many FDA-approved medications And worldwide, there are so many government-approved medications for COVID-19. Number one, D is dexamethasone. It's a steroid, and that was confirmed to have mortality benefits with a recovery trial by Oxford University. Number two is remdesivir. Remdesivir is the only antiviral approved for COVID-19, and it's very effective in shortening the duration of COVID-19. Some institutes combine another JAK1 and 2 inhibitor called baricitinib to remdesivir, but remdesivir is given across the board in this country. Antibiotics, A for antibiotics and ancillary vitamins, very important. Antibiotics, why? Although it's a virus, it's associated with high mortality when there's secondary pneumonias. So we give ceftriaxone and azithromycin in that situation. Vitamins are vitamin C, vitamin D3, and we also added zinc sulfate to that. And A for antibody cocktails, I think Dr. Jayaweera is going to talk about new exciting antibody cocktails that will prevent hospitalizations in high-risk patients with mild to moderate infections. Eli Lilly and Regeneron both put out these monoclonal antibody cocktails. We got the FDA approval for that. And P for plasma, convalescent plasma. Initially, it's going to boost your antibody pool to combat this virus. P for proning. So if you sleep on your sides or on your belly when you have shortness of breath, that'll help optimize your oxygenation. And last but not least, E for enoxaparin, which is a blood thinner, anticoagulant. So timelines for the development of various vaccines. So I'm going to dive right into this now because obviously we are talking about vaccine. It is very exciting. And this is completely unparalleled to what was going on before. We are about to witness the greatest mission that the US government and other governments undertook last century. What is that? Rollout of a vaccine or vaccines. Now, when you talk about vaccines, we're going to talk about two different types of vaccines: traditional convention type of vaccines versus fast-track emergent type of vaccines. As you can see on this slide, polio was the classic traditional vaccine. It took about more than 60 years to develop an effective polio vaccine. Salk and Sabin, right? Sabin oral vaccine actually eradicated polio from this world in 1981. So polio is no more except in 3 countries: Afghanistan, Pakistan, and Nigeria, for very well-known reasons. But Ebola vaccine is somewhere in between. Ebola, it came into limelight in 2014 with a massive pandemic. Unfortunately, we didn't have the vaccine. But the same thing happened in 2019, 18 or 19 in Congo. We had the vaccine at that time. So that actually prevented a lot of death because Ebola, as you know, can be transmitted through blood contacts and blood-related contacts with 100% case fatality, mortality, right? So that was prevented by the VSV vector vaccine called Ervebo, E-R-V-E-B-O. Now we are talking about 3 different pandemics, coronavirus pandemics: SARS, MERS, and COVID-19. Those are typical pandemics, right? Those pandemics have to be dealt with therapeutics and vaccines. Unfortunately for SARS and MERS, we didn't have a vaccine. There were preliminary attempts to come up with a vaccine, but because of lack of funding and other political issues, those attempts died down. But COVID-19, hats off, ever since the Chinese scientists gave us the genetic code for COVID-19 in January, a lot of universities, academic centers, and companies across the globe jumped to the idea of coming up with a vaccine. Now, these were well funded by 2 major organizations: Operation Warp Speed in the United States, and that's a government-sponsored organization or alliance, and then WHO. We're going to talk a little bit about that. So ideal vaccine development is given on this cartoon, and courtesy Dr. Jayaweera. So ideal vaccines developed with a rigorous process, as you know. Research and development and validation of components of the virus and the targets. Once you are done with that, you jump into preclinical animal studies. So I'm going to go through this very fast. So once the preclinical studies are over, we're going to jump into clinical studies. Phase 1 about safety. It's all about safety. Once you've passed that, you go to safety and efficacy. Phase 2. Efficacy is how effective the virus is. Now we have two vaccines. Meduna Vaccine, Fisa Vaccine, followed by Meduna Vaccine, both 95% effective and almost 100% safe. This is a great thing. This is actually the greatest achievement on earth last century or so. Phase 3 clinical trials, they are going to look into efficacy, protection of COVID-19, and then the other intricate details. So once you go through that process, we need to find out whether the review, the other scientists' reviews and the logistics are looked into, manufacturing, worldwide distribution, those things. And supply chains have to be addressed. We have come across a huge barrier in supply chain because we had to maintain ultra cold supply chains, ultra cold chains for Pfizer and Meduna vaccines. So typically, we don't use human challenge studies for vaccine development traditional way, but in pandemics and in certain situations, we have used human challenge trials to fast track phase 3. Otherwise, we'd be stuck with 5 to 10 to 15 years of vaccine development. Rather than that, new technology, the cutting edge technology has helped us to go through this process of phase 3 very quickly so that a vaccine can be designed and developed within a matter of 9 to 10 months. So when you talk about vaccines, we have to talk about adversity. It's all about safety, safety, safety. Adverse reactions, and side effects. If you develop adverse reaction side effects, you're not left, in the dark. And you have to know that you're not alone. We have some very, very strategic systems in place to help you out, such as a smartphone app called vSafe, which will be sent to you by CDC. vSafe is a smartphone or smartphone-based tool or app. And there's another a system called Vaccine Adverse Event Reporting System. It's already in place, actually, even with other vaccines. It's called VAERS, V-A-E-R-S. And then the provider can actually report adverse events and the side effects right away. If you are injured, if you are hurt with the vaccine, there's a way to get compensated. It's called Vaccine Injury Compensation Program. That's also in place. And then for healthcare workers, which is the first layer of COVID-19 vaccination, Axion Health platform is already in place. So, but before talking about the vaccine, we have to, be very grateful to these two organizations, COVAX, followed by Operation Warp Speed. COVAX is an alliance of vaccine development. This is the vaccine pillar of ACT Accelerator of WHO. So this was launched by WHO, Gates Foundation, and CEPI, which is Coalition for Epidemic Preparedness Innovations. And the whole point of putting together this alliance is to make sure that there's fair distribution of this vaccine. Not only that, but also to support the countries and the companies who were struggling with funds in creating this vaccine. So the goal was 2 billion doses of COVID-19 vaccine by the end of 2021. More than 1.5 billion pledged for global accessibility and fair distribution of this vaccine, thanks to the efforts of WHO and 184 countries in that. Operation Warp Speed was set up by the US government. It's a government initiative to mitigate the financial risks of vaccine and therapeutic development by providing funds to the companies and helping coordinate clinical trials. In other words, Operation Warp Speed has given money, funds to the pharmaceutical giants to go ahead and help out the scientists to develop the vaccine. Not only that, they will make sure that the distribution, the rollout will be done smoothly, and everything's in place as we speak. The first Pfizer vaccine was was launched a day before yesterday. That was December 11th in the United States. Pfizer vaccines are already approved in 6-7 countries already, including the US, UK, Canada, Bahrain, Saudi Arabia, and Mexico. So all these are in the US. The entire intricate and complicated, convoluted process of vaccine distribution is monitored by Operation Warp Speed. And this is a coordination between CDC, NIH, National Institute of Health, and Biochemical Department, Department of Defense, Biomedical Department, Department of Defense, and DHHS, that's Department of Health and Human Services. And basically, the very first company, pharmaceutical company that benefited from Operation Warp Speed was Johnson & Johnson. In March, they got $450 million for vaccine development, and they're doing a great job with that. And Dr. Jayaweera is going to talk about it. He's actually in the thick of it. Other partners such as Pfizer, Moderna, AstraZeneca, Regeneron, Sanofi, and GSK, and convalescent plasma, Mayo Clinic, all those benefited from Operation Warp Speed. So when you talk about vaccines, there were so many vaccines about 6 months ago, kind of racing against each other, racing against time. So when you talk about the preliminary vaccines, Pfizer vaccine wasn't even here. Pfizer vaccine, which is an mRNA vaccine, made a beeline to the front thanks to the dedicated efforts of all the scientists they working for them. But on this slide, you can see Moderna vaccine, Moderna is a mRNA vaccine. Moderna company in collaboration with NIH, the United States NIH, actually designed this study in July, launched the phase 3 trials, and now they have the results. And interim analysis showed that it's 100%, it's 95% effective. So Moderna was there from the beginning. The most important thing is that mRNA concept was used against Zika, for Zika virus vaccine, but unfortunately Zika epidemic died down, so that was given up. But as you can see, there are some other vaccines, University of Oxford on this slide, which started off work a few months ago, along with Moderna. And there was Johnson & Johnson. You can see that there's Sanofi one with Institute of Pasteur, and then there's an Indian vaccine. Of course, Serum Institute of India is the biggest vaccine in the world. They were there from the beginning, and they are focusing on a live attenuated virus based vaccine. And there's a Chinese one called Sinovac, which is using an inactivated virus platform. So as you can see, there are so many platforms that they use, but primarily for COVID-19, we use two platforms, mRNA platform by Pfizer and Moderna. That's a brand new concept, but we have had tons of experience with that, with Zika. But second one is even more time honored, and we have better experience with the second one called viral vector vaccine. These viral vectors that we use to produce vaccines are non-replicating, non-disease causing. The commonest viral vector that we use, adenovirus, right? So Oxford used an adenovirus to produce their vaccine, and they are getting very close to get it approved by the regulatory boards. Johnson & Johnson also used adenovirus 26, and these have been used in other previous epidemics. So diving right into Pfizer vaccine, Pfizer got the approval from FDA through emergency use authorization 2 days ago. So the biggest rollout on earth, the biggest rollout in the United States history of a vaccine will happen on Monday. That's tomorrow and day after. And the very first person will get the vaccine. And after that, we're going to vaccinate about 20 million people, primarily phase 1A healthcare workers, phase 1B nursing home patients and the nursing home workers. So administered in 2 doses. We all know that now, 3 weeks apart. And it's approved for 16 years and older. So there's a bit of a controversy about vaccinating 16, 17, 18 years, which is kind of like a waste in my mind. But so because of that, there are some negative votes on the FDA panel. But finally, they came to the agreement that we need to approve this right away, right now, real time. It is an mRNA vaccine. It's a brand new concept, but it's a very safe platform to make vaccines. It's an easy platform to make vaccines. 95% efficacy. Vaccines are all about safety, number one, efficacy, number two, and durability, number three. So 95% effective is a great thing. And this was proven with a phase 3 trial done among 46,000 volunteers. And those volunteers are not just healthy volunteers, but also they have about 40% of diverse backgrounds. So that tells a story. The side effects, so safety, side effects that people experience during these trials were minimum. That's a great thing for us, right? Minimum side effects such as injection site pain, fevers, headaches, like a little bit like a hangover effect, they say. So if you take the vaccine today, make sure that tomorrow you have a relatively light day. That's what they say. Unexpected anaphylactoid reactions were noted in 2 people in the United Kingdom on their V-Day, vaccine day, last Tuesday. And that is very, very common, actually. It's not common, but it is accepted as a adverse reaction for any new medication or any new vaccine. And mind you, these 2 people had the background of anaphylactic reactions to other medications. They were carrying their EpiPen. So again, I don't think that's a big deal. This is the very reason why we monitor patients or people for 30 minutes, at least 30 minutes after we vaccinate. So folks, the most important concept here is not just the vaccine, but also vaccination. If the vaccination is done with the due process being followed, this kind of things can be avoided. Transportation and storage is a huge issue for Pfizer because it needs an ultra cold, chains, right, for transportation and storage, minus 70 Celsius. So the real question is, can those other countries afford to buy minus 70 freezers, ultra deep freezers? Now, in my hospital, we bought a couple of freezers. It cost about $15,000 a freezer. But there are some hospitals in the United States who are not, wealthy enough to buy, which are not wealthy enough to buy these freezers. So a way out for them would be a thermal boxes. We're going to explain later. So the logistics part of it is that the distribution is via thermal shippers. So Pfizer had designed these specific thermal shippers with GPS-enabled thermal sensors. That's the most important thing. So this will make sure that the temperatures inside will be maintained between minus 60 and minus 80 Celsius. In Fahrenheit, it's about 90 to 100. It is stable for 10 days, unopened, stable for 5 days if open. So we are only allowed to open those boxes twice a day for 90 seconds, right? The box must be returned at the end of the 30-day period. If stored in the refrigerator, it's stable for 5 days, as we explained before. But another disadvantage with Pfizer vaccines, it needs dilution. Once diluted, stable at room temperature only for 6 hours. It's a very short time, So those are the logistic difficulties that we come across with this vaccine. Ultra sub-zero freezers cost about 10 to 15,000, and McKesson is a supply giant. They have already shipped the kits with syringes and alcohol pads and the face shields to all the sites. And then UPS and FedEx, as well as American US airlines, have already done their duty to transport these heavy boxes of vaccines from Belgium to Michigan to the rest of the country. Meduna Vaccine will be the next. Next week, we'll have Meduna Vaccine FDA approved under EUA, I'm quite sure. It's a LNP encapsulated mRNA vaccine. It encodes for a form of S protein of the virus. It's developed by Moderna and NIH, as we pointed out before. Phase 3 trials had 30,000 volunteers. Typically, we require only 3,000 volunteers for EUA approval. But these vaccines had 30 to 60,000 volunteers. I think Johnson is targeting 60,000 volunteers in their trials. This is also administered in 2 doses, 3 to 4 weeks apart. Efficacy and safety data from phase 3 announced November 16th, very similar to Pfizer vaccine, and 37% were from diverse backgrounds. So Meduna Vaccine logistic parts, 94% effective in preventing, as I said before. This trial was called HOPE trial. This was designed even before the Pfizer trial. No side effects were found. 20 million doses will be have been projected by the end of the year. That is 10 million people. And common side effects are injection site pain, headaches, fatigue, muscle pain, and stable at refrigerated temperature. We're going to talk about this when we answer the questions later on. At refrigerated temperature, this is stable for 1 month. That's very important. Frozen for 6 months. You get stable in frozen temperatures 6 months. So it's more thermal stable than the Pfizer vaccine. And another advantage, no dilution required at point of care. But unfortunately, this has now been tested in children and pregnant mothers. So they were very upfront with that. Pfizer tested them in some children. So that's why they approved for 16 years and above. So Johnson & Johnson, I think Dr. Jayaweera is going to talk about it. This is the only vaccine which is given in 1 dose, and it's a thermally stable, so it's suitable for warmer temperatures. That's using adenovirus platform, which is a viral vector platform. Oxford vaccine in partnership with AstraZeneca. Again, it's a time-honored platform. It's a viral vector platform called chimpanzee adenovirus OX1, CHAD OX1, adenovirus virus vector, and very safe that way. There have been just one side effect of the phase 3 trials called transverse myelitis, weakness of lower extremities, but that was monitored and cleared by the regulatory bodies. So the phase 3 trial is still going on, and I'm quite confident that in January we'll get the approval for the Oxford trial. But the good thing about Oxford vaccine, is this can be manufactured in other countries. If you read India's websites, they've already undertaken manufacturing, and Australia. They've already undertaken manufacturing the Oxford vaccine, millions of Oxford vaccine. For India, 600 million Oxford vaccine within the next 3 to 6 months. So that's very doable in other countries. And they are working in close partnership with WHO and Operation Warp Speed. This is the cartoon or illustration of the viral vector-based vaccine development. From one side, on the left-hand side is the chimpanzee adenovirus. So that's a virus that is unable to cause disease because it's unable to replicate. From the right-hand side, you take the spike protein of COVID-19, and we do some gene sequencing, genetic coding, spike protein, and gene sequencing, and then we combine the two together to form the vaccine. And this vaccine can generate neutralizing antibodies. I'm also very excited to talk about Sanofi and GSK, Glaxo COVID-19 vaccine. Unfortunately, there's a bit of a delay just a few days ago because of the lack of efficacy in older folks, but they're going to correct that. As you know, Sanofi is a pharmaceutical vaccine giant, and over the years, it has produced so many very effective vaccines, including your influenza vaccines. There's a French company of Sanofi, Pasteur, influenza vaccine, hepatitis A, B, varicella, and Bordetella, and so on and so forth. So they use a platform called protein-based platform. So Sanofi and Glaxo use a protein-based platform. And then the next one that I'm going to talk about, Novavax, are also going to use a protein-based platform. So these are a time-honored platforms, but that use it's an adjuvanted recombinant technology. So it's a brand new, like cutting-edge technology to produce 200 million doses as they pledged to COVAX and WHO, and 100 million doses pledged to Operation Warp Speed. 2 doses, 28 days apart, just like the other vaccines, except the Johnson & Johnson vaccine, which is only given in 1 dose. Phase 3 clinical trials will be, they were targeting to end these phase 3 clinical trials by the end of January, but it's not going to happen in January because of the hiccup that they experience in older population, unexpectedly low efficacy because of low antigen content. So the second Sanofi virus vaccine is made in partnership with Translate Bio company, which is an mRNA vaccine. So stay tuned for that. Novavax is a protein subunit-based vaccine. Novavax is a US company, and it is also using an adjuvant a supportive chemical called Matrix M. Matrix M has been used in the production of NanoFlu, the brand, another new quadrivalent influenza vaccine. So it has history and experience. 2 vaccines 3 weeks apart. Phase 3 trials already had 15,000 volunteers in UK completed. So those results will be out very soon. 25% were over the age of 65 years, and 5,000 were tested in South Africa already, and some of them were HIV patients. So we're going to get some new perspectives with Novavax trial. So it is using nanoparticle technology, nanotechnology. And Inovio technology is also very important because this is a very fast track trial. And this is one of our neighbors, San Diego Labs. They recently announced they can make a vaccine within 3 hours. This is a DNA vaccine, and DNA vaccines can be produced very fast, very rapidly, whereas, in 3 hours. That's a very short time. It's in phase 3 clinical trials. And actually, MERS vaccine, the only MERS vaccine that was in phase 1 trial was produced by Inovio. And it's fast, stable, and quick designing and manufacturing process. And safety profile is and tolerability profile is so good. But the thing about Inovio is it is delivered directly into the cells via a smart device, a proprietary smart device. Now, we don't know the details about that at this point. So having said all these things about potential vaccines that are going to come out within the next 3 to 6 months, I always want to point out that there are about 200 vaccines that are, the phase 1 trials are going on at this point. There's a lot of information about vaccines. A lot of people are getting confused with these vaccines and the data and information about vaccines. So it's important for us to sit down and read about some of the latest data and news about common vaccines. So I would invite you to read about Fisa vaccine, Meduna vaccine, Johnson & Johnson vaccine, and Oxford vaccine. Those are the 4 front runners. When you talk about, last but not least, when we talk about vaccine development, we cannot forget the fact that safety is our primary goal. Now, there are 2 syndromes that could potentially be caused by a new vaccine. Something that we experienced with dengue virus vaccine called antibody-dependent enhancement. So this could actually create a cytokine storm. So it's important to be aware of these things. There's another syndrome called vaccine-associated enhanced respiratory disease. These are very, 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 very uncommon side effects of vaccines. Again, last but not least, I'd like to thank all the healthcare workers who have put their lives on the line in saving lives. And this is not going to be easy for all of us. Next 6 months or so, we will continue to be struggling in saving lives. So far, we've taken care of hundreds of thousands of patients in this country as well, and world over. So hats off and salute to all the healthcare workers. At this point, I'd like to introduce Dr. Dushyantha Jayaweera, who's a professor of medicine and infectious disease specialist at the prestigious Miller School of Medicine at University of Miami. Over to you, Dushyantha.
So first of all, I want to thank the Alumni Association of University of PERAHERA Ottawa chapter, and for putting this webinar together along with the Sri Lankan Foundation. You can't mention Sri Lankan Foundation without thanking Dr. Walter Jayasinghe and his wife, and also Deepthi, who introduced me to the foundation, and I've been working with them closely on these webinars. I want to thank Shirani for that introduction. Wow, my mother couldn't have introduced me better. That is really something. Thank you, Shirani. And of course, Anura, for giving me some tips on about the slides about the audience, because I think if I gave my original plan, I think that would have been not very suitable. And Her Excellency, Madam Ambassador, what she said was very true, that, you know, it's actually along with the challenges that one gets with things like COVID-19 comes the opportunities for people to spend time and, you know, catch up with their families. Unfortunately, that didn't happen to me and Deepthi because I haven't taken a day off since April. I have worked every day, Saturday, Sunday, and weekends because we are running clinical trials, we are seeing patients, so it just doesn't stop. So with that, I get started, and I'm particularly fond of PERAHERA because my older brother was the dean for the medical school many years ago, and he passed away 2 years ago. So I have been to PERAHERA many times. With that, I'll get started. So I kept off the vaccines because I can't say anything more than what Deepthi has already mentioned about vaccines. So, but I'd be happy to answer questions. But I left out the vaccine part on my talk. So I come from the HIV world, as Shirani mentioned, that I am done research on HIV for a long time. And I had to be careful when I say I come from the HIV world. Last time I said that, somebody texted me and saying, hey, I didn't know that you had HIV. So I had to say, no, I do HIV research. Not that there's a huge difference. The life expectancy change between the HIV and non-HIV is about 7 years. And if you're going up to about 90 years, I mean, 7 years is not a huge deal. But the important thing, I put this slide on the plague because this I used to think when the HIV came. But this was written by Albert Camus about the plague in North Africa. And one of the greatest sentences was they fancied themselves free, and no one will ever be free so long as there's pestilence. And this is one thing that we, when I say we, the Sri Lankan, the middle-income countries and low-income countries were always aware of the pestilence. And there is an inbuilt plasticity in the health system, always expecting pestilence to come. And the freedom, so people mistake freedom as, you can talk what you want and do what you want. That's the United States definition of freedom. But I think countries like Sri Lanka understood what freedom slightly differently. Freedom is the freedom of health, education, and those things, which we had got it really right. So despite our wealth, technology, and scientific knowledge, the current pandemic, the deaths have been the greatest among the weakest living in the strongest countries. I think there was a question in the chat line, I don't know why Africa is so protected. We'll talk about this later. But I think what really showed was that small countries have more plasticity in the health system in the presence of pestilence, and you can move things around. So with that, I can get back to something what we're going to talk about. So I'll give a little bit of introduction, and I'll talk about a little bit of epidemiology, how the disease, how each countries are reacting. I'll talk about Canada, Sri Lanka, and other countries. The clinical presentations, I'll talk about the diagnosis and treatment, and we can take some questions after that. So I like this thing, your Alumni Association, the emblem, which says "Together we march forward," which is actually very true. So as of today, just this 2 hours ago, I checked the Johns Hopkins website, and Sri Lanka today has got 32,000. It's really sad because we had a really good system in place for prevention, and we just lost it. I have the plan. I'll show in a couple of slides down, but still, compared to the rest of the world, we had done pretty well. The deaths were only 149. Florida, which has the similar population to Sri Lanka, has 1.1 million, despite how rich Florida is, and we had 19,000 deaths. As Deepthi said, his group has had 5,000 cases. I work at University of Miami and Jackson Memorial System. We have about 8,000 cases over the pandemic. We have managed. Fortunately, that our death rates have been pretty good in our system. Across the world, you see this, 71 million, and Canada has done reasonably well at 458,000 cases with only 13,000 deaths. So Canada has, I think, managed it quite well for a developed country. So if you look at the daily new confirmed cases, I put this graph to show relative incidence about the cases. Obviously, Sri Lanka was doing very well at the beginning, and but then we had these spikes. part of the problem is that, you get, COVID fatigue. You can have the best system in the world, but if people don't follow instructions, this happens. And then, we are human. We all make mistakes, and then that's what it is. And so I hope that Sri Lanka will attend to this and get this under control. And this, again, the cumulative number of cases. You can see if you everything is relative in life. So if you compare, I don't know how, by mistake I clicked Russia here when I was downloading the data, but Sri Lanka, Canada is right at the bottom, and of course United States has a dubious distinction of being highest in the world. Obviously, I don't want to get political, but you all know what went wrong with the United States. Then if you look at the tests conducted per case diagnosed, so because the prevalence is low, Sri Lanka has to do more tests to pick up one case. I don't know much details. This is coming from Johns Hopkins and Oxford. We have doing more testing to pick up cases, but we are doing a good job, and it just shows that we have the right attitude. So we just got this paper published between myself, Walter Jayasinghe, who is the professor of medicine in Peradeniya, in Colombo, in the American Journal of Public Health. We basically addressed the COVID-19 in a resource poor setting. We basically was trying to highlight that if you have the leadership, then you can come up with some really innovative ways of controlling things, and you don't need so many resources, and you can actually do things. The Vietnam has a tremendous advantage, and my wife is from Vietnam, so I have strong connections with Vietnam. Vietnam had this singular the advantage because they have been exposed to the H1N1 and other pestilences and epidemics. So their system is well geared to dealing with pandemics. It just shows how resilient they are. The second thing is that their culture is that they wear masks, like the Japanese. If anybody in Vietnam has a cough or cold, they wear a mask. They go on the bicycles and motorcycles, they wear masks. So the mask culture is built in there, almost like in their DNA. So it's easy when you say wear mask, everybody wears mask. Nobody is saying, oh, I like freedom, my personal freedom, I'm not going to wear a mask. That nonsensical talk doesn't exist in Vietnam. So that's one of the reasons. And social distancing. So this was the plan that developed. Mind you, this was developed in end of January. Nobody was even thinking about this. Nobody was even thinking about this in other countries. When this was developed by the Sri Lankan government with help from the military intelligence and the health, and they actually brought up all the experts in the country. And they brought the military intelligence because they knew how to track things. So this was, this is published in the web, so this is nothing secret, this is all, everybody knows what I'm talking. And so they came up with the fundamental public health measures. This is what you teach in the medical school. Actually, they are taught in high school now in US. You have simple detection and isolation and tracing for a pandemic. So they use the big data analysis, they use the data cross-checking, record cross-checking, and the ground intelligence that people going and checking on people. And they focused on highly vulnerable communities, moderately less, you can see the picture. Isolation, self-quarantine or central quarantine with curfews, village isolation, lockdowns, and again, tracing, using cell phone technology and all that. So you can see this was done in February. So we managed to control this to a long time. And then there was a breakdown in the system. But it's not due to lack of understanding the gravity of the pandemic or the lack of understanding of what to do. It is just the human errors. So this was the system that they used. If there was an index case, then they will go and look at the data. The index cases were picked up from reporting data, immigration, telephone technology, you're tracking on your cell phone usage, hotel reservations, and different reportings. And they tracked down the contact tracing so that they could follow it. And once the detection was done, so they did the confirmed cases, then sampling of associates of positive cases, random sampling of high-risk cases, which US is doing now, and so sampling at border control checkpoints, which they did quite well. And they were using the PCR testing, and if you are positive, then they obviously followed them, and then they're negative, they're left alone. The importance of the PCR testing is that their false positives are less and the false negatives are less. So there are other tests like antigen detection, and those tests have a lot of false positives. So as much as you want to pick up these things, having false positives is not a danger to the public, but it's a danger to the individual, because it's just imagine that if you're false positive and you have to go through all the quarantines, you can go to work and all these complications, and it's not just fair by people. So you have to try to minimize false positives. So that's so what I'm trying to say is that you have to be proud about Sri Lanka. How well they dealt with it, despite our spikes now, that it just demonstrates the clear thinking of Sri Lanka. So now, before I get to coronavirus, I want to just separate this off, coronavirus and influenza. So influenza has a two-breed group called influenza A and influenza B. And so the coronavirus, on the other hand, is a different virus. Coronavirus leads to respiratory distress syndrome, was first identified in 2003 in South China, and then it came back in 2012, and then of course COVID-19 was diagnosed in 2019. Coronavirus causes severe respiratory problems. So as a result, people die before it spreads. At least the SARS and the MERS. COVID-19 was slightly different. It was like a lower grade of infection. It does cause death, but not the way SARS or MERS were causing. On the other hand, the influenza is much more benign, although it kills a lot too, but it's not the same rate as corona. And in 2009, there was the novel coronavirus H1N1 pandemic, that because of the antigenic shift completely from H1N1 to the pandemic flu, which was quite devastating. So there's a fundamental difference between these two. So now, before I get to, Deepthi talked about the vaccines and how good the Moderna and Pfizer are, and even for when Oxford is coming, their vaccines are slightly less. But if you look at the flu vaccine, it's only about 45% for any influenza vaccine success, and 51% among the young, and the 37% among the old. So this is the success rate that we are currently vaccinating people. So when coronavirus vaccine comes, it is 3 times more effective than flu vaccine. So it's unconscionable that if you refuse to take this vaccine, of course, people are worried about the safety and things like that. Unfortunately, in the United States, the general public has this tremendous suspicion about these vaccines because of some crazy people making conspiracy theories. So in general, in the United States, only about 60% people agree to vaccinate. Now, let me compare this with Sri Lanka. Sri Lanka, according to WHO, you can Google it, 99% success rate in vaccine. So in fact, it was too good to be true. I called my colleague, Walter Jayasinghe, asked, can you just check the data? Because Sri Lanka had 99% success rate. Because one thing is that we have an infrastructure that is well developed for community care. We have medical officers of health, we have community case workers, we have the village headman. In fact, United States is trying to do that. They are starting a new thing called community health workers, which we have been doing it since 1950s. So you have to see that we have systems well placed in Sri Lanka. So if vaccine comes, Sri Lanka would be vaccinating almost 100% if we could afford it. So I'm going to skip this slide because Deepthi showed it. So the question is, so how long does it take once you get infected? How long does it take for you to have symptoms differ. That's what we call the incubation period. So the incubation period is 5 days, roughly about 5 days. This is coming from the Chinese data, and this has been reproduced by multiple studies in US. It's roughly about 5 days, but the duration may vary. There's of course a standard deviation. It from quite a lot. I mean, it could be a couple of days to 3 weeks, but basically incubation period is 5 days. And these are the symptoms. So you can come with headache, you can come with cough or sore throat, shortness of breath. We all know that. But the funny thing is that now people are coming with atypical presentations. So we are wondering whether the virus is slowly drifting antigenically, or our previous exposure is changing us or the virus. Something is changing because, and for example, I was doing one of my regular clinics on Friday. I picked up 2 patients. One came with saying, I have diarrhea and I feel tired. No shortness of breath, nothing. So I said, hey, listen, I need to test you for COVID-19. So I had to send him off and, isolate him. The second patient came with, I lost my appetite and I can't smell. Obviously, that is but unless you ask, people can say, I cannot smell. But if you directly ask the question, they say, I cannot, my smell is going off and my taste is going up. I don't feel like eating. So it's important to pick these things up so that you can take them away from the transmission, because fortunately these people, we don't bring anybody without a mask on, and then we are wearing masks, and I don't wear a face shield these days, but I wear a N95 mask, so and a cover, I'm probably safe. So it is important to be aware of atypical presentations of COVID, because it's not everybody who comes with shortness of breath. So the symptoms may appear 2 to 14 days after exposure to the virus. So generally, there's an incubation period, which I said 5 days. So it plus or minus then the symptoms appearing. So mean duration of symptoms are about 11 days. So once you get it, it lasts about 11 days. Medium time from the first symptom to death is about 14 days. Now, the death rates vary from place to place, and of course, depending on the comorbidities. As if you're older, like then you die faster. If you're over 70, it's about median time is about 11 days. If you're younger, it takes longer time. So, but fortunately, in most academic centers and most good centers, the death rates are very low, very low. We have managed to, we hardly have any deaths in our academic system. Yeah. And these are the symptoms that people, as I mentioned before, headache, loss of smell, nasal obstruction, asthenia, just tired, cough, muscle aches, runny nose, taste is fine. If you just have a runny nose, that doesn't mean that you have COVID, but, you just have a cold, or, something wet up your nose. But other than that, you have to be careful not to overthink, but when in doubt, get tested. So WHO has classified these symptoms into, there's a thing called ordinal scale. I didn't put it because I don't want to complicate life, because you guys, I know that most of the audience is not a medical audience. So, but I want to keep things simple. I think, you said, keep things simple. So I'm keeping it simple. So there's the mild illness and the moderate illness and the severe illness. Mild illness, and you have a little bit of cough, little shortness of breath, but you have a pulse oximeter and you check your oxygen in your blood, 90 saturation, and it's over 95%, so you just stay at home. Moderate is that when still oxygen is O2 assay is greater than 95%, but there are clinical signs of chest x-rays showing that things are not good. Severe illness when the oxygen saturation drops less than 94%. And the O2 saturation, you might say, oh my God, 94% is pretty good. But the thing is that the oxygen dissociation curve, for you guys who have done chemistry, it's a sigmoid curve. So if you drop your O2 saturation from 94 to 90%, the partial pressure of oxygen drops tremendously in the blood. So that's why that we keep at 94%. And then, you don't need all these fancy things. You can just check the respiratory rate and how the patient feels, and the things are not good. I grew up in Sri Lanka. I went to Sri Lankan Medical School, and we were trained to practice medicine under a tree. So I mean, I could do this without any of these fancy gadgets. So critical illness is obviously, if you're in shock and respiratory failure and multi-organ dysfunction. So now let's get on to the testing and diagnosis. So the WHO defines a suspect case definition as a patient with acute respiratory illness, fever, or 1 or 2 symptoms. Now, obviously, WHO needs to change this a little bit because things are changing. Obviously, and then of course, history of travel to a residence. I mean, this definition needs to change because now you don't have to travel anywhere. You go to the supermarket, you can get it. You can go to a restaurant and you can get it. You go to church or temple, you can get it. In fact, I go to COVID units almost every day because I am doing a large convalescent plasma study with the National Institute of Health. And sometimes my coordinators, who are quarter of my age, scared to go in. I said, Lynn, if you're scared to go in, I will go in. So I would go into COVID unit and talk to the patient, get a consent. We can't take the paper out of the COVID unit. So what we do is that we take a picture and we email it to my team, and then my team will print it, and then we put it on our regulatory documents. So I am actually more comfortable going inside the COVID unit than going to a restaurant or go to the doctor's cafeteria because everybody is eating with the mask down, and I'm more worried about that. In fact, I think most of the people feel the way. So the method that we use is RNA. Unfortunately, what is happening is that RNA takes, doing a PCR, doing method, a polymerase chain reaction to looking at the RNA, finding the RNA in the sputum or in the swab that we take from the nose or the nasopharynx, or if the patient is intubated, we take a lavage from the lungs. So any of those, and there are other methods of doing, like now. What happened in our unit was that we were getting so many tests, our lab couldn't keep up doing PCR because our PCR machines are all packed. And then we started doing antigen detector called Acula technique, just looking at antigen. The problem with the antigen detector is the antigen detector gives false positives. So it's a huge problem for me because I'm running research, and then my study says that I need a PCR test positive, and if I do Acula test, it doesn't confirm. So I have to somehow get the PCR test done. So this is what the gold standard is. And it's funny, we think about the sputum and the swabs from the nose as the gold standards, but actually there's a fair amount of COVID in the stool. There is a similar viral load in the stool, and in fact stays longer. So we could actually check stools, but it's more difficult to get these and process, and the PCR machines are easier to do with preparation. But if you miss on the in the sputum, you can always run on the stool PCR, and you can pick it up, and it stays longer. So that's the important thing. So one would ask, like, why is it in the stool? Because as Deepthi said, the angiotensin II receptors are in the nose, in the mouth, in the throat, in the bronchial lavage, bronchial mucosa, in the alveoli, but is also on the intestinal tract. So the COVID-19 could enter through the GI tract into the body. Right? And so these are important aspects that you have to remember. So now let me briefly take you through, like, how these things, how this over a time sequence. So if a virus, I have the slide that Deepthi showed, where the virus comes in and enters and gets into the ACE2 receptors and the viral replication. So this just tells you how step by step what comes positive. So if you exposure it at day minus 2 weeks, and day minus 2 weeks, and then once, say about 10 days before the symptoms come, the nasopharyngeal swab will be positive. So if you had exposure in a couple of days, even before the symptoms come, if you do a nasopharyngeal swab, the PCR will come positive. Then the next one was the virus isolation of respiratory tract. That also peaks, but the respiratory, it goes down quite significantly from the respiratory tract if you take it from, if you're supposing you bronch the person, it comes down very quickly. And then sorry, I made a mistake. So this is actually from the just coming from the respiratory tract. Bronch is the one that lasts longer. Bronchial lavage or sputum PCR. Then if you look at the antibodies, so the IgM are the early antibodies, stays shorter. IgG is the longer antibody, stays, comes later, but stays longer. So the IgM antibodies comes just before the symptoms, and the IgG comes a little later, and then it stays longer. So this is so the test what you do has to be based on what time the things started and what time the things are going to end. So, I mean, if you do antibodies too early, you're not going to get anything. But if you do the antibodies, and if they're positive, that means you have been exposed at some point to the infection. And if you do the PCR after 4 weeks, likely negative, and you can't say that I never had COVID, because in that case, if you run an antibody, your antibody would come positive, but the PCR is negative. So that's the thing. So, and somebody actually once asked me, like, what's the best time to get the second vaccine, or what happens, how does the immune response work? So I put this graph. Okay. So if you look at if you expose to antigen, whether it's a vaccine or an infection, the primary immune response take about, as I saw the IgG and IgM, IgG goes up and it starts coming down. But if you get exposed to again, now you get a huge second response, right? And so this is what some people ask, like, okay, so if I had COVID-19, do I need a vaccine? It depends. It depends. Now, if you had COVID-19 and if the infection lasts very long, like, if you're in ICU for 28 days, you have a pretty robust immune response. Most likely you don't need a vaccine, but you can always check your IgG levels and consider that. But if you had a mild infection, then you may benefit from a vaccine. In fact, I'm doing the Janssen vaccine, as Deepthi said, and we accept people who had COVID-19 before, and if they were after 2 weeks, we will put them. So we take a baseline antibody titers, and then we vaccinate, and we want to see how the antibody titers would change over time, second to vaccine. As Deepthi said, our Janssen vaccine is single dose, so it's easier to manage. So somebody asked me, like, how long does the stay effective once you get the vaccine? So we don't have data on COVID vaccine because, I mean, let's face it, Moderna started in early August. Pfizer started around the same time. Janssen started late September, and there was a 2-week hold. So we don't have anything. So we had to go by H1N1 vaccines and the flu vaccines. So in general, I'm just giving general, how long does it? So roughly about 150 to, the vaccine efficacy is about 150 days or maybe 180 days. So let's say 6 months. And then as the infection goes down, the influenza risk keeps going up. So this is the deal. So this is one of the things that we talk about, like, if you have somebody who want to protect for influenza, and if you get the vaccine in, say, early, like, say September, then if the winter lasts longer, towards the end of this winter, that you may be at little higher risk again. So especially if you're elderly, you need to take the high dose vaccine. At least the data shows, I mean, data is not completely solid, but general consensus is that it's better to give high dose vaccine, flu vaccine, to keep this going longer than 180 days. So we may have more information as the data is analyzed for Pfizer and Moderna. And I don't think, I mean, obviously, mRNA vaccine and a DNA vector vaccine are two different, completely two different techniques. So we need to see how each one will work. Hopefully they both work very well. So the treatment. So Deepthi already spoke about this, so I'm not going to too much detail about this. I think one of the things about treatment is that we have to get away from taking into theoretical possibilities and making them into big factual stories. When I was preparing for, I mean, not preparing, but writing this down, I realized that most of the things that I said in March were all wrong. And in fact, we are taught in the medical school that 50% of the things doctors say are wrong. The problem is we don't know which 50%. So when the doctor says this, you have to ask, well, let's analyze what you say and let's look at the data based on what you say. And so that's another thing I teach medical students, that to be humble. You have to be humble because we are wrong 50% of the time. So you cannot say that, oh, because I have an MD degree and I wear a white coat, what I say is absolutely true. No, it's not true. What we can say is that the best case scenario, this is what we think based on what we have now. And then the things may change over time. I'll give you a good example. When COVID came, hydroxychloroquine, you know the story about hydroxychloroquine. Everybody was going crazy about hydroxychloroquine. And I myself thought, well, I looked at the basic principles and I found that hydroxychloroquine had an antiviral effect and also anti-inflammatory effect. So we thought, well, theoretically this should work. So, and there are some, I was working with a colleague at the National Institute of Health, and they have these drug development platforms where you have these in vitro assays, and you can throw a drug into the in vitro assay and see whether it works or doesn't work. So to do this, you have to have a lab which is biosafety level 3, which is like, where you work Ebola and stuff like that. So not many places have such a lab. And hydroxychloroquine works. But the reality is that when you try it on patients, we were one of the first to study that called a HERO study, health exposure research outcome study, with the Duke University and the University of Florida. Myself, we were doing it. And it was first we were doing for prevention, but there were 10 studies done for treatment. It didn't work. It didn't work. So there were so many things. Antacids were tried, it didn't work. Colchicine was tried, when it didn't work. I can go through about, angiotensin receptor blockers were tried. Theoretically, if you block the angiotensin receptor, you could stop the virus from entering, but that doesn't work. In reality, it doesn't work. I mean, the reason why some of these things work is that we think that the viruses are as dumb as humans. The virus is actually pretty smart. So, for example, HIV enters the cell through the CD4 receptor and a chemokine receptor. So we, in 2000, we developed a chemokine receptor blocker. So we thought, okay, if you block the chemokine receptor, the virus can't enter. 2 days later, the virus decides to come through another receptor, and the whole thing crashes. So you can't generalize some of the theoretical possibilities to clinical practice without going through the steps and really finding. So the simple things work. Oxygen, that works because you don't have oxygen. We give oxygen. We found that the people are having a lot of strokes and cardiovascular disease. It was almost like inflammation inside. In fact, now we are finding, the CDC just reported on the MMWR, that after COVID is over, after about 4 months, people start developing inflammatory reactions. Some people have blurry eyes, some people have difficulty in smell, some people have joint swelling, some people have cardiovascular events, some people have heart attacks, some people have strokes, and they are all related, and some people have difficulty in remembering. So you check this and say there's an inflammatory reaction. So one of the things that we're finding was when COVID came, there are a lot of people having blood clots. So blood thinning works. Of course, mechanical ventilation. Deepthi went through this, I'm not going to go into details. One of the things was prone position. If you flat on supine, face up, there were more chances of lungs collapsing than if you put your face down. So that works. And antacids, if needed. So the convalescent plasma, it's a nice story. Convalescent plasma has been given by Mayo Clinic over 100,000 people. And they just didn't bother to do a study because we all got carried away saying that we have to save lives. So the one thing we learned from the pandemic is that as researchers, our academics, we have 2 roles to do. One is to save lives, but you have to keep the research going. You cannot sacrifice randomized clinical trials just to say that I need to do the best for my patient, because sometimes what you think is the best is not the best. So therefore, that we made simple mistake by not doing a randomized control trial of clinical convalescent plasma when the when the pandemic started. So as a result, the Mayo Clinic spent almost millions and millions of dollars giving this plasma to everybody in US without any evidence that whether it works or not. So we are running a style with the NIH and the Operation Warp Speed randomized control trial. So we have now gone up to about 600 patients. We need 1,000 to exactly know whether it works on people who are hospitalized. So I'm the principal investigator for Miami, and I don't know who gets what, but we need to do it. So we still don't know whether convalescent plasma works or not. So I think Deepthi talked about remdesivir as antiviral agent. So it's interesting that any remdesivir is an antiviral. It was first developed for MERS and COVID-1, and it theoretically works in test tube. And I already mentioned to you that test tube doesn't cut it. Just because it works in test tube doesn't mean that it works. It stopped the RNA activity. So theoretically, it's possible. There are 3 studies that were done. The problem with these studies was some of them were not randomized. Some of them were not controlled for severity. And they didn't control for concomitant medications. These are fundamental principles in clinical research. You have to control for this. So the Infectious Disease Society of America gave remdesivir approval only for the people who are severe COVID, patients in room air on or supplemental oxygen or mechanical ventilation. Only those 3 people. So the reason is that there is not enough data. So we have to be very careful about these drugs and the pandemic situation. We mustn't get carried away. I remember when in April, May, June, July, August, there is a drug called tocilizumab. It's a IL-6 blocker. It's an interleukin-6 blocker, because what I think Deepthi was talking about, the cytokine storm. What happens is that when the virus enters, it disrupts the cell system that in our bodies that controls infection. Instead of controlling the infection, it causes chaos, absolute chaos. And we thought that if you take out one of the chaos players, IL-6, that we could settle everything. And then we get fooled by the pharmaceutical companies you should never trust. That they say that, oh, I got the drug, we can block IL-6, and you're a fool not to use it. Actually, we were fools to use it because without a study. And after some time, we realized that tocilizumab doesn't work. So we spent billions of dollars on that for something that never worked. So that's how the Western world health system spends too much money with very little outcomes to talk about. So of all these drugs that you're talking about, now here is this Infectious Disease Society of America looking at the drugs and telling, okay, this is what we recommend for you to use. This is the first column is the people who are at home. Here are the people who are mild. This is the severe, and there's critical. So the 4 columns, mild, the not hospitalized, mild, severe, and critical. If you look at hydroxychloroquine, it says don't give it to anybody. Azithromycin, don't give it to anybody. The HIV drug, lopinavir, ritonavir, don't give it to anybody. Because there were so many theoretical possibilities, and the in vitro assays which show that these things work. Don't give it to anybody. Then corticosteroids, that was done by a study from the UK National Health System ran the study. They said that it must be used on critical people. So now that's solid evidence. Dexamethasone should be, if you can't afford dexamethasone, give any steroid. Dexamethasone, daily dose of alternate glucocorticoids. For hospitalized patient, you can suggest, it does not recommended, but can use. The problem with this there's a saying in life that too much of a good thing is a bad thing. So if you overdo these things, what happens is that if you give too much dexamethasone, you make the person more prone to infection. So you may get rid of COVID, the respiratory problem. Downstream, you end up with fungal infection or something like that. The same thing that happened with tocilizumab was IL-6 blocker. There is a reason that IL-6 is there to control it. If you block it, downstream, you end up with fungal infection or something like that in the ICU, and the patient dies. It's like this old story, like operation was successful, but patient is dead. I mean, you don't want that to happen. And then remdesivir. Remdesivir is recommended in basically hospitalized but not severe. This is the one that the hospitalized critical patients may benefit, and these are so basically randomized only for the hospitalized patients, right? It's recommended, but the WHO didn't recommend it. Basically, WHO is looking at the broad countries like middle income, low income, and say, wait a minute, if your healthcare budget is X, I mean, you're not going to spend all your money on remdesivir. Yeah, there are much better things to do than that in, say, in Sri Lanka or India or Vietnam. I mean, you could give basic health care, basic ICU care, and be pretty effective. Now, as Deepthi said, these monoclonal antibodies, Regeneron antibody, which is being suggested for mild to moderate cases, even the Infectious Disease Society says, be careful, don't use it routinely. Even, in fact, recently I had one of my colleagues' father was infected, and they asked me whether they should use this drug, and I said no. As long as he's feeling well, don't use it. Just because you have it, don't use it. Wait till you need it, and then we'll use it, because the data is not solid. So that's why I was, I think when I was talking to Anura and them before the talk, that I said that, it's very important that you face what is really evidence-based rather than, what people say. So if you're going to give Anaphylactoid, then of course, there are infusion criteria. So you really have to have, you have to be obese, you have a kidney disease or diabetes or immunosuppressive, if you're over 65, you have a whole host of things before you use monoclonal antibodies. Now, for example, I think we are told that the president got it and he got monoclonal antibodies. Rudy Giuliani got monoclonal antibodies. Well, I think they have practically everything that is in this list. So I'm not surprised. So now, with that, I think I have come to an end, and I want to thank you for inviting me to speak, and I'll be happy to answer any questions. And I want to thank again the article that I got from COVID-19, the Sri Lankan publication, Sri Lanka Foundation Leadership, and of course the Alumni Association of University of Peradeniya. Deepthi, who it's very hard to follow Deepthi after his talk, so but I'm trying. And thank you very much, and I'll stop now.
Thank you so much, Dushyantha. I learned so much from you. Every time you come up with a presentation, such a wonderful, such an insightful presentation. For the, as we're running out of time, we're going to jump right into the questions. Can you mix the vaccine doses? Such as from two companies, two platforms?
So this I get this question asked every day. So I have this standard answer, because I have, if you're going to mix, I mean, first of all, we said, please don't mix. Why would you mix? I mean, if you're getting Pfizer, they're not going to give one shot. They're going to give two shots. If you're going to go with Moderna, you're going to get two shots. If you're going to go with Janssen, they're going to give one shot. Now the question is, if you get Pfizer and if you mix it Janssen, we strongly don't recommend. We would say that talk to your primary care. Very unlikely any primary care physician would recommend that because we do not know what the interactions could be.
Right. So the second question, can mRNA vaccines cause autoimmune diseases in patient populations?
Very unlikely. So the way the messenger RNA works is that the messenger RNA, so this is a novel concept by Moderna and BioNTech. So the fundamental theory was that messenger RNA is like the coding that tells, it doesn't get into your DNA. So if you give the messenger RNA, it gets, so you need a way of getting the messenger RNA into the cell. Once it goes there, what it basically tells the, if you do remember your high school biochemistry, like, Golgi apparatus and all the stuff that manufacture proteins, messenger RNA gives the coding to the endoplasmic reticulum, endoplasmic reticulum and Golgi, tell that, okay, I want you to make a protein. So a protein that is reflective of COVID-19. So a lot of people think that, these things will change your DNA, that you will become like, oh, some bionic person, that in fact there was somebody was saying that MIT has put some coding that they can track you, and the mRNA gets removed immediately. So we don't know whether the autoimmune diseases could occur. That is something that people are following, but it is unlikely.
Right. Thank you, Dushyantha. So next questions about RNA and DNA virus, which is more virulent. We all know that there are certain DNA viruses. Yeah, go ahead. I'm sorry.
Yeah, so the virulence has nothing to do with the RNA or DNA. For example, DNA virus is papilloma virus. It's a big virus, it doesn't, cost very much, except you give you a little wart. RNA virus is the HIV. Can kill you. And in fact, everybody long ago thought that every virus was the DNA virus. In fact, the guy in 1968 said that there is the RNA virus, and he was laughed out of the conference saying that there's an RNA virus. And 10 years later, he gets the Nobel Prize. Because people said, how can you have RNA virus? Because RNA, for to replicate, it has to cause a reverse transcription and a DNA copy to get into the nucleus. So we don't know anything about that, but he was right. So there's no difference between RNA or DNA about the virulence. Hepatitis C is RNA virus. So the DNA viruses, there are some other infections which are not. There's no connection between RNA or DNA and the virulence.
Right. Common is RNA, DNA virus that we come across, that we deal with on day-to-day life with patients are like herpes virus, right?
Yeah, exactly. Adenovirus is the virus also.
Those are common viruses, but they are all all over the map. So there are no much difference as far as disease-causing ability or virulence concerned. This is a good one. If you have had COVID-19, should you get the vaccine?
Yeah. So this is the point. As I said, 50% of the things I say is rubbish. So we don't know. So if you had a severe infection, you would have developed a really robust antibody response. And very unlikely you need a vaccine, because at the end of the day, when you do a vaccine, what you're doing is that you're trying to fool the system by giving something of the virus and making the immune system recognize that, oh my God, you got COVID-19. So if you got the real thing, why would you want to go and give a vaccine? The flip side of it is that, and then of course, once you get the infection, it's kind of in your slide, Deepthi, you showed like, you have the neutralizing antibodies, IgG, and also the cellular antibodies like B cells, which have memory, which will recognize anytime something else comes in. So if you get an infection, you already have that. But the problem is that if you get a mild infection, if you look at the IgG titers, they keep going down after some time. So there are a lot of people who have had infection, we go and test them, they have no antibodies. But still, they have a cellular component, which have B cells and T cells, which will probably be able to respond in the event they were exposed. So in general, we have said you don't need it, but I tend to tell that if you have a mild infection, there's no harm in getting it, taking the vaccine.
I agree with you, Dr. Jayaweera. So this is how we break it down for people. This is a very common question that we get asked. So the answer is we don't know, but most likely you will after about 2 to 3 months, because based on some of the studies published already in New England Journal of Medicine, the classic one was Imperial College London. They published a study, small study, but they showed only 16% of the antibodies that lasts beyond 3 months. So if you get the infection, mild form, chances are you don't have lasting immunity, T cell or B cell, you better get the vaccine. If you have had a very serious infection, we are going to have a different conversation, right?
Yeah.
So answer is, you don't
exactly.
know. We recommend everybody to get this vaccine, but if you have the infection, at least wait for 3 months. Next question would be, if you are an asymptomatic patient, if you're an asymptomatic patient without symptoms, should you take the vaccine even if your antibody titer is high? Because we know that asymptomatic patients, minority, like a small group of people, minority of people can have unknown infections in the past, and their antibody levels are high. Now, those antibodies are not going to be, we don't have the quantification. We don't quantify the antibodies, unlike in academic centers. For people, for commercial basis, we cannot tell you that this is the safe level of antibodies, such as hepatitis B. If the hepatitis B antibody titer drops below 10, we say go get a booster. So based on that, I would say we don't have quantification or quantity-based PCR at this point. Therefore, you would know whether this antibody will last long versus it'll die short.
So so I agree with you. So say I'm doing this convalescent plasma study. So what we do is that before we give the convalescent plasma, we quantify the titer. So we want the dilution 1:320 dilution in the convalescent plasma. So most of the studies that were done with the vaccines, what they did was they gave the vaccine, you pull their serum, they look at the antibody titers and compare with the convalescent plasma. So what they found was that if you cannot match the convalescent plasma, the vaccine does not work. So the answer to the person who asked the question, as you said, we need to quantify. Just having a yes or no answer does not cut. If you quantify, and if your antibody titers are more than 1:320, then you have to think, I think I look good there. But you're right. I mean, we will know more answers in another 6 months. If we have a seminar like this, we will have more answers.
That's right. So folks, this is the most important question that you guys have been asking us. After vaccination, vaccination is going to happen within next 6 months for everybody. Phase 1, phase 2, phase 3, and phase 3. Phase 3 and phase 4. Now, after vaccination, can we relax our safety guidelines? The answer is no, because we are still going to be in the middle of a pandemic. I talked about 95% efficacy for some vaccines, and other vaccines such as Oxford vaccine, 70% efficacy. So we are talking about symptomatically positive, symptomatically active patients being tested for COVID-19, 5% for Moderna and Pfizer, and then maybe 30% for Oxford vaccine. So we're talking about only symptomatic. If you don't have symptoms, you're not going to get tested, right? But there's a big area called asymptomatic infections. So you can still get the vaccine and get asymptomatic infection. We don't know. In phase 3 trials, they didn't look into, to my knowledge, they didn't look into asymptomatic infections. There's a small chance that you can get still infected. You're asymptomatic, so that you won't be tested. So bottom line, as a corporate statement, I would say, yes, you still have to wear a mask and mitigate all the mitigation steps that we discussed. Continue with those things, including the distancing, hand washing. That's going to make your health, your hygiene, and the world's hygiene better, no matter what you say, right? Please take care of yourself in the hospitals as well as outside. Anything to add to that?
No, that's it. At least in the Janssen, what we do is that we see them 6 times on the first year, and we take a swab, and we have picked 1 or 2 asymptomatic cases, but most of them were within the first week before the vaccine could have any beneficial effects.
Right. And I saw another question. I can locate it at this point. This is for Dushyantha again. If you have had penicillin allergy, can you take the vaccine? Or peanut allergy, can you take Pfizer vaccine?
Yeah, I would be very careful. I would be very careful. I mean, so there are, I mean, you have to tell your healthcare provider and make sure that your primary care physician is well prepared. I would be ready if, I mean, I don't know, because so you have to be very careful if you have serious allergies, let's say like peanut allergy or things like that.
Right. So that's what that's another common question we get asked in the private sector as well, in our hospitals as well. So if you had serious allergies requiring EpiPen to be carried by you, you're not going to take the vaccine, right? And one more area is if you have had major neurological complication due to a flu vaccine, such as Guillain-Barré syndrome, like weakness, ascending weakness, you're not going to take these vaccines, right? Then you'll be on roll. Continue the mitigation trial. So those are the relative side effects that are relative contraindications that we. So remember, if you have had serious allergy to anything, medication, food allergies, you're not going to take this vaccine. If you had Guillain-Barré syndrome, you're not going to take this vaccine. Another question: can COVID lead to depression as a complication of disease? So we've explained these post-COVID syndromes over and over and over again. Once you have had COVID, 14 days, you will have symptoms or asymptomatic. But post-COVID, after COVID, a lot of patients are coming up with this chronic fatigue syndrome, brain freeze, something that we call myalgic encephalomyelitis, right? Your whole body is aching, arthritis, arthralgias, right? Pain in the joints, and then rashes, and then loss of smell, loss of taste, lasting longer time. And then among kids, there's a syndrome called Kawasaki-like syndrome and depression, right? So all those are relatively uncommon, but very well defined in the post-COVID studies. Any validity that BCG gives some immunity? This is a common question for all of us, right? Yeah, go ahead, Dushyantha. BCG. Right?
Yeah. So about depression, I mean, we have had studies where we show that inflammation is directly related to depression. There are a couple of NIH protocols we are running for people with depression, treating for inflammation, and there's some reasonable results. So I think what you said was absolutely true, that the post-COVID syndrome as depression we have seen. That's what was the BCG. The BCG, that's a long story. So so this was based on the observational data that showed that the countries that gave BCG had a lower incidence of COVID. And then one of the questions somebody asked about Africa, why Africa didn't have that much COVID. One of the reasons people gave was that, plus that the social distancing was much greater in Africa because there's not much, people are not bunched together that much, and also the exposure to other pathogens gives you have some kind of immunity. But BCG is a hard question, and the Germans were running a clinical trial, and there are a couple of clinical trials in clinicaltrials.gov website. Some people are running, so it'll be interesting to see what it shows.
That's right. University of Melbourne still running their BCG clinical trial. University of Queensland actually failed with their clinical trial, just announced, because they used the HIV protein to produce a vaccine. But unfortunately, some of those patients developed HIV antibodies. So they didn't develop the disease, but they developed antibodies. So because the mistrust created, so they pulled out their vaccine. So another question, Pfizer vaccine, my understanding, if it only prevents the disease of COVID, but still you have COVID virus in the body and could potentially transmit. Can you please comment about the Pfizer vaccine?
No, I don't think so. I don't think so. I mean, basically, it prevents. I mean, the whole idea of the immune system is that when when the virus comes in, it gets neutralized, and so the virus doesn't get a chance of establishing disease. So it prevents. It's not that. I mean, all the vaccines does the same thing, So I don't think it gets asymptomatic infection by vaccine allows asymptomatic infection.
Another timely question: is there a chance that COVID-19 could mutate? Yes, we know that we've had, I think, documented 6 mutations now. Now, currently we are going through this surge that we call a second wave. Now, this is not rocket science. All the time we predicted, we forecast a second wave. It's going to come when there's cold weather outside, such as in the West and America, the Western countries, including America. But I have a feeling that this could be a different strain because so many people are coming in. Part of the reason must be the compliance issue and then the behavior issue because of the holidays that we experience. But sometimes it's kind of beyond our comprehension as to why so many people are getting affected. So these are not, or the replication factor, how contagious the virus is. And I've lost faith in these numbers because I'm sick and tired of taking care of these patients. I'm tired. I'm actually fatigued. So, and I'm pretty sure Dr. Jayaweera will say the same thing. especially badly about, with the minority communities such as Hispanics and African Americans. I don't know whether a mutation is responsible, a new mutation responsible for this enormous surge. What do you think, Dushyantha?
I think there is some degree of antigenic drifts. It is the problem is that one of the challenges of studying this is that, as I earlier said, that we need the biosafety level 3 labs to study these things. And even in the United States, there are only very few BSL-3 labs available to actually sequence the virus and see whether there are real changes. I mean, we have viral samples, but we don't have a lab to do these things. So it is possible that there are some changes happening. It's hard to say. Yeah, as you said, that we cannot explain why this keeps going up and up and up, despite all the precautions we take. It is true that people don't listen to advice we give not to. The thing is, people get fatigued by just fatigue that's doing the same thing over and over again, just like all of us,
Yeah, you have to understand, ladies and gentlemen, we are just two infectious disease specialists who've been taking care of thousands of patients, but we still don't know about the real answers to some of these questions, right? There are so many unknowns out there. So obviously these answers might change if we have another platform, Zoom platform discussion 2-3 months time. Another important one is side effects, short-term side effects, long-term side effects. A lot of people are worried about long-term side effects, especially with the DNA vaccines such as Inovio, right? Is injected with a smart device. So obviously those are valid questions. The big answer to this is we do not know. Short term, we can tell it's a very safe, those are very safe vaccines. Long term, I can only tell you this. We have taken so many vaccines in our lives, including the childhood vaccines, adult vaccines, such as, some of y'all would have received pneumonia vaccine, influenza vaccine we receive every year. So what are the long-term side effects of those? We don't know. Well, you cannot be 100% sure about this and live a normal life. So you have to trust somebody. That your mother told you, right? You have to trust somebody. That's what it is. So I will trust scientists and take the vaccine because this is the only exit strategy from this pandemic. Thank you. So anything to add to that about long-term side effects? No. All right.
Thank
you. Another question is, I don't know, we are running out of time. So should I, Keshini, I think I had to ask Keshini, should I give up on these questions or answer them later on? Another interesting one is, can the virus enter the body through conjunctiva or cornea, through the eye?
Yes.
Yes, you can. That's the reason why we are wearing face shields and eye goggles in the hospitals. Basically, universally, we do that now. Every hospital worker are required, taking care of patients are required to wear a face shield for that reason. And another question about DNA vaccines. So can the DNA vaccines change your genome for good?
No. You're just introducing a small DNA sequence. It is not going to change. This is the conspiracy theory that people trying to say that at least to our knowledge, it is not true.
That's right. So another question: if President Trump and Rudy Giuliani got the antibody cocktail, monoclonal cocktail, how come it wasn't given to young people who were dying of this disease? Again, it comes to the moral conundrum, so to speak, right? Dr. Jayaweera will address that. He did address it. But go ahead and answer that.
Yeah.
So you want to add to that, Dushyantha?
No, just because both of them, they are not like role models for anybody. So just because they got it doesn't mean anything, because sometimes it's a VIP syndrome. When you're a VIP, they try to overdo things. I'm sure they would have got better without them also, because the trials didn't show that And also that Rudy Giuliani and Donald Trump, they were, I think they got mild infections. So that's the population that monoclonal antibodies would work. So they didn't get a severe infection.
So, so I'm gonna take one more question. What's the difference between mRNA platform and DNA platform? Remember, I explained to you there are common platforms we use. So, over to you, Dushyantha.
So the messenger RNA platform is just the messenger RNA. It's actually a really novel technique. The DNA platform has been there before. DNA, the difference is that the mRNA, you have to get into the cytoplasm, the cell, inside the cell, not the nucleus. Whereas the DNA platform has a way of injecting the DNA into the DNA. So you have to have another piece to introduce the DNA into the nucleus. So it's a little bit more complicated.
That's right. Yeah. So I think we're gonna stop at this point, and I'd like to thank once again. I cannot express enough gratitude to Keshini and Chamod, Shirani, Dr. Walter Jayasinghe, and Aisha Jayasinghe for organizing these events. Now we have a new partner in crime, Ottawa chapter of the Canadian PERAHERA Medical Association. Thank you very much for organizing these events. We are learning as much as you are learning from these Zoom talks. And hopefully within next month or so, or a month or 2 months, we'll come up with another talk or a Zoom meeting. Over to you, Keshini, to wrap it all up.
Such great presentations, a lot to absorb. I forgot what I was going to say right now too. But anyway, I'm Keshini Vijay Gunratna, the Director of Project Management and Public Relations for the Sri Lanka Foundation. I always like to start out by thanking our foundation team, Dr. Walter Jayasinghe, the founder, Dr. Dushyantha Jayasinghe, the president, Aisha Jayasinghe, the vice president, Shirani Stanislaw, the chief operating officer, Jayaweera Singhe, the director of cultural affairs and director of Academy of Performing Arts, Lal Tilakaratna, the principal. Last but not least, Chamod Amarasinghe, the media director, who takes the leadership to run the webinars behind the scene. Thank you so much, Chamod. I am here today following the vision that Dr. Walter Jayasinghe put in place to connect with our communities, to bring awareness, to educate during good times and bad times. Unfortunately, this is one of those bad times. With that being said, I'd like to extend my sincere gratitude to our heroes, the doctors who put their lives at risk to save others and their families at risk as well. Dr. Deepthi, I'm so sad to hear how tired you are, honestly, and you look tired. You look exhausted, so does Dr. Dushyantha. And thank you. You even took the time to come through to our communities to do these presentations, because I know you haven't had any days off the past few months. We've conducted about 5 webinars in the past, I guess, 8 months. These are very first time collaborating with another country. It has indeed been a great pleasure working with the president, Dr. Nimal De Silva of the Alumni Association of the University of Peradeniya, Ottawa, Canada chapter, and his executive committee, Dr. Dambika Herat, Manoj De Silva, Dr. Anura Herat, and Sampath Henayakam. It has indeed been a great pleasure, and thank you so much for working with us. Thank you to all of the attendees, all of the special guests, and looking forward to see you all at the next webinar. Now I hand over the webinar platform to Dr. Anura Herat. It's all yours, Dr. Herat.
Thank you, Keshini. Actually, you have done my job partly, but this is really fabulous. I mean, we have learned a lot, and 2 doctors, our thanks goes to 2 of you, and you are simple in presenting your facts and knowledge, but very, very effective. And we have had, I think, more than 100 participants from all over the world. And interestingly, I think all of them stayed on, even, the participant from Sri Lanka it's about midnight now, but they stayed on. That showed how interesting your presentations and the facts and how useful those would be. So thank you so much for sharing your knowledge and being so generous in presenting your knowledge to everybody. And so from our side also, I would like to thank again our organizing committee. And I must tell you all that this was very, very tremendous collaborative effort and also all voluntary efforts. And so therefore, I would thank our organizers, Dhammika, Manoj, Sampath, and Deepani for putting everything together, and Keshini from your side, and also Chamod. And also I would like to thank again our sponsor, Erika, and for helping us to build up our funds a little bit. And it was a great pleasure having all of you join us. And we had about 100 participants, as I mentioned. And I wish all the best for you, and stay safe, and also stay tuned for our next set of webinars. Thank you very much.